mentioned in class this morning, we got two things we're doing today. One is going to be inoculating a variety of different media with your your two knowns and your two unknowns. And a lot of these are just looking at the metabolic capacity of each of those four organisms, telling you about either their ability to utilize a particular substrate or to produce a particular byproduct. And there's two basic mechanisms for indicating their status. One is to use a pH indicator. So many times when a microbe is capable of utilizing a substrate, as a result of that utilization, it's either going to produce carbon dioxide, which carbon dioxide in solution causes the pH to drop, right? Or those organisms are going to ferment that substrate and produce some sort of acidic byproduct, so some sort of organic acid like acetic acid or lactic acid. And so adding a pH indicator to a media that has a unique substrate allows you to determine whether the microbe can use it. Right? It's very straightforward. Also, one other metabolic byproduct that is often detected by a pH indicator is ammonia. So often when microbes are utilizing amino acids as their primary energy base, they're going to produce ammonia, and that's going to actually alkalinize. It's a great word, or increase the pH, and so you can see that as well often with the pH indicator. And that should be familiar. That idea was discussed when we talked about the sulfur-indole motility media. Utilization of cysteine or tryptophan could also lead to alkalinization, which would be, in theory, detectable. So we have a couple of different pH indicators that we're looking at here today. One is bromothymol blue, which is going to span a yellow to blue color depending on the pH. That's utilized here in the OF glucose. It's also utilized down here in the citrate utilization media as well. Phenol red broth, which uses the pH indicator phenol red, pretty straightforward, which spans yellow to kind of a magenta color depending on the pH. Methyl red, which actually has the reverse coloration depending on the pH. So it's red and yellow, but it goes in the opposite direction. Those are all pH-based indicators, right? So they're all looking at the ability of a microbe to produce some kind of end product, usually an acid or carbon dioxide. So how do we actually use that in these different contexts? So probably I'm actually going to start with the phenol red broth because that's probably the most straightforward here. So with phenol red broth, in the broth, there's very little in there to begin with. You have what we call phenol red broth basal media. And basal media means it doesn't have any sugar in it in this context. So you can buy phenol red basal media, and then you can add whatever sugar you want to test. Okay, so in this case, we chose two sugars. We chose lactose and sucrose. Both of these are disaccharides, and not every microbe can utilize those. Okay? So if they can use lactose, right, then the lactose, phenol red broth with lactose, will change color in response to the utilization of that lactose if they're producing either carbon dioxide or an organic acid of some sort, like lactic acid or acetic acid. These phenol red broth tubes also have another unique component to them. They have inside of them these little things called Durham tubes, which are just an inverted tube inside of here, And if the bacteria are doing a fermentation that's resulting in the production of a gas like carbon dioxide, you'll actually start to accumulate gas inside of this inverted tube. Okay? So this phenol red broth tells us whether the organism is capable of fermenting a sugar to produce an organic acid of some sort, right, either lactate or another organic acid, and whether they're producing CO2 as a byproduct. Okay. We're testing two different sugars, lactose and sucrose. These are both disaccharides, both of which are going to be, the disaccharide will be split, and then you'll have some sort of isomerization to convert each of the monosaccharides into glucose, right, glycolysis to pyruvate, and then pyruvate fermented to some sort of organic end product. Any questions about that general concept?
So which is the gas that's indicated within that other tube?
So if you're getting gas in here, like 90% of the time you're going to get CO2.
Okay.
There, in theory, you potentially, I suppose, could produce some other gases, but most of the other gases that would be produced are either not produced in sufficient quantity. So like cysteine, if you were removing the sulfhydryl group from cysteine, you could in theory produce hydrogen sulfide, but it probably wouldn't be enough. There wouldn't be enough produced to see. Other microbes will produce hydrogen gas or methane, but that's typically in an anaerobic context, and these are not anaerobic. So CO2 is basically what we're seeing if we see gas being accumulated. All right, then moving on. The second one's very similar concept here, but we're looking at glucose specifically, and we're looking at whether organisms can oxidize and or ferment, right, glucose. And this is a fairly straightforward system. We have a media which has a very high sugar content. In this case, the sugar is glucose. It has a very low peptone, and I guess I probably talked about it before, but just to clarify again, peptone is a digest of a protein substrate of some sort. Okay, so it's taking pepsin and digesting meat, usually, right? Some sort of beef byproduct. So this would be a source of amino acids and things like that. So there's very low peptone in here, high sugar content because we want them to be preferentially utilizing the sugar if they're capable of doing so. This does have a low amount of agar in it, in part to make it a little bit easier to work with, but also to minimize oxygen diffusion. We saw this with the fluid thioglycolate media as well, right? We put a little bit of agar in there to minimize the diffusion of oxygen through the media. This is a little bit higher agar concentration than FTMs, but it's not like what you would see in a plate. Okay? How do we get some degree of differentiation between oxidation and fermentation? We actually are going to inoculate for each organism that you're testing. You're going to inoculate 2 tubes. One of them you're going to leave the top open, and by open, I still mean putting the cap on, but you're going to leave it open as opposed to the second tube, which you're going to put a layer of mineral oil on the top. And that layer of mineral oil is going to keep oxygen from getting to this media. Okay? So after you inoculate the 2 tubes, one of them you'll put a layer of mineral oil on. Usually a ml should be plenty. And if the organism is capable of utilizing glucose under these 2 different conditions, again, you're going to see either the production of an acid via fermentation or the production of CO2, which will also cause a drop in pH, and we'll see some yellowing Excuse me. So an organism that can ferment, right, can ferment glucose is actually going to give you a positive in both of these tubes. Right? Fermentation doesn't require oxygen, but it can still happen even when oxygen is present. Right? So an organism that's capable of fermenting is actually going to give you a positive in both of these tubes. If an organism is only capable of oxidizing glucose, you're going to get a positive here, right? And you're not going to there. If it can't do either one, you're not going to see a color change in either context. So think about that as you're interpreting the results tomorrow. Which ones turned yellow, and what does that actually tell you about the metabolic capacity of each of your organisms? Any questions about that?
Are we using all 4 organisms or both of them?
Yep. So all of these tests today, you'll test all 4 of your organisms. So you'll need 2 tubes for each organism here, so you'll have a total of 8. You'll need 1 lactose and 1 sucrose phenol red broth for each of your 4 organisms. And they are so handily labeled by our wondrous TA, Anna. So the phenol red broth lactose have red caps, and the phenol red broth sucrose have clear caps. So you'll need 4 of the clear and 4 of the red today for that one. All right. The next one is called the MRVP, which stands for Methyl Red Vaux Proscour. Again, I always wish that they'd stop naming things after people instead of telling you more about what's actually going on. In this case, you're going to inoculate a tube that has glucose and peptone in it, so it's sort of a general nutrition source. It has both some sugar and some protein base, plus a few other things that come along with the peptone. After 48 hours, so it is important to give this one a little bit longer. After 48 hours, and it could be even longer than that if you need to, you're going to split this into 2 tubes. So you can leave some in your initial tube and just transfer a couple mils out to a second tube. And one of the tubes, you're going to add methyl red, and the other tube you're going to add Barrett's reagents A and B, which are also sometimes labeled VP reagents. So these are here labeled VP reagents, right? And the basic idea here again, methyl red is a pH indicator. So if the organism is producing what we would call mixed acids, you're going to get a really bright red. If it's orange to yellow, that's a negative for the methyl red test. The VP portion, if you add the Barrett's reagents A and B, you need to wait just a little while. This is testing for the production of a non-acidic fermentation end product, which is called acetoin. And acetoin is basically going to react with alpha-naphthol, which is one of the Barrett's reagents, and then it's going to coordinate with guanidine, which is in the peptone already, and it'll give you a nice bright red color here. So both of these, you're looking for a red color as a positive for this test. So 48 hours, split it, the methyl red into one of the 2 tubes, and then the Barrett's reagents, A and B, added sequentially to the second. Any questions about that? The next two tests are actually looking at, really looking at oxidation of different compounds, or the capacity to oxidize things using some sort of electron transport chain. So we'll start by looking at catalase here a little bit. When we talked about organisms' tolerance for or utilization of oxygen, right? We talked about facultative anaerobes and obligate aerobes and obligate anaerobes and microaerophiles and all those kinds of things. One of the things we didn't really talk very much about at that point was why oxygen can be a problem and what microbes do to tolerate it. So with most electron transport chains, at the end of the chain, you need to have a molecule that can receive the electrons that came through the electron transport system. And for our mitochondria and for a lot of bacteria, that's oxygen. Okay? Now, when that happens, typically the oxygen is going to receive electrons sequentially, and so it actually can happen in kind of a two-step process, and if it receives some electrons and manages to kind of escape from the terminal reductase, you're going to generate some superoxide. And superoxide radicals are very, very reactive. They'll oxidize anything. Like they'll react with just about any kind of molecule in the near vicinity, lipids, proteins, whatever. And so it's very, very damaging. So most organisms that are utilizing oxygen are going to have a couple of enzymes to help deal with that superoxide that gets generated. So the first one is called superoxide dismutase, which is going to basically react superoxide with water and with hydrogen, and we're actually going to end up generating some hydrogen peroxide and some oxygen. Okay? Now hydrogen peroxide itself, what context do you use hydrogen peroxide in? What? Yeah, disinfecting things, right? So hydrogen peroxide itself is very good at killing a lot of microbes, right? So it itself needs to be dealt with, and so many microbes will also have a second enzyme called catalase. We have catalase actually, right? Like in our blood, which is why if you ever put hydrogen peroxide on a wound, right, the hydrogen peroxide is going to decompose into water and oxygen. The bubbling that you see is actually oxygen gas being generated. So this is the same thing, same idea. To perform this test, it's very simple. You're going to take a slide. You'll put a loop full of your microbe to be tested, the broth. Put a loop full on the slide, and while it's still nice and liquid, you add a drop of hydrogen peroxide. And if you get bubbles, it's positive for the test. Okay, fairly straightforward. So, right, the main thing we're really looking at here, right, is this presence of catalase, which is really an adaptive feature of microbes that are utilizing oxygen or utilizing some sort of electron transport chain where superoxide can be generated. Any questions about that? All right. The last one, the oxidase, second to last one. The oxidase test, this is very simple to perform as well. This is really looking at the presence of cytochrome C oxidase. So this is a particular component of some electron transport chains. So not all organisms that have an electron transport chain will have a cytochrome C oxidase. We'll talk more about differences in oxidases in coming weeks in class. But this is testing for a particular oxidase, the cytochrome C oxidase. We're testing for it using a reagent called tetramethyl p-phenyldiamine. And this molecule is sensitive to the redox status of the cell broadly, but more specifically, it's capable of contributing electrons to the cytochrome C oxidase. And if it does, right, it will become oxidized and it will change from a colorless to a purple color. So there's two different systems for doing this test. One is to utilize these little tubes which have the TMPD in them. You can crack these and they put a little bit of sample on a slider and a spot plate and then add this to it. The other one, which is simpler to use, is called the dry slide, and it kind of looks like a 4-pane window, and each of the 4 panes can be used to test a separate organism. So if you get one dry slide, you can test all 4 of your organisms using that. Okay. Questions about that?
We call for move full of our organisms on one slide, or we should use separate slides?
Just use one slide, but a separate window for each of the 4 organisms.
No, I mean like number 5.
For number 5, yeah, you can use a clean slide for each one. It'll be fine. All right. The last one here is the citrate utilization test. So citrate, if you think about metabolism as a whole, when we talk about respiration, right? We have the citric acid cycle, right? Citric acid, citrate is the anion, right, of citric acid. So you would think that any organism that's doing sort of metabolism like we do in our mitochondria would be citrate positive, but that's actually not the case. And the main issue is that not every organism can take up citrate from their environment. Okay, so in this test, we're not really testing for the capacity of the microbe to utilize citrate. We're testing primarily whether the organism can take it up from the environment. So we're testing for a specific protein called citrate permease. Okay, so this is a transporter for citrate to allow it to get into the microbe. To some degree, we're also dependent on the presence of a second enzyme called citrate lyase, which can convert citrate to oxaloacetate, which can be converted to pyruvate as part of the normal sort of central metabolic pathways of the microbe. So, but the main idea that we're really getting at is whether or not the organism can take up citrate. How are we detecting it? We're actually detecting it using a pH indicator again, but in this case we're mostly looking for an increase in pH. In this media, the only carbon source is citrate. Okay? In addition to that, about the only other 2 things in there is some ammonium phosphate and some bromothymol blue. If the organism is utilizing citrate and trying to grow and perform sort of normal metabolism, it's going to utilize this ammonium phosphate as well and produce some ammonium ions and also some ammonium hydroxide, both of which are going to drive the pH up. The bromothymol blue, this is the same bromothymol blue that we were talking about over here. So the citrate agar is going to go from kind of a greenish color to a really bright blue color if it's utilizing citrate. Okay. These are poured as a slant, so do the drag, right? Drag inoculation with your loop, but should be pretty straightforward. Any questions about that? All right. So like I said, this week is mostly about utilizing a pH indicator of some sort to indicate something about the metabolism of the organism, either its ability to utilize a sugar, something like lactose or sucrose, or its ability to produce a particular end product, like mixed acid fermentation or a neutral fermentation end product like acetylene. And then sort of the second theme is looking at something related to the oxidative capacity of the microorganism, something related to an electron transport chain, utilizing either the catalase test or tetramethyl p-phenyldiamine, phenylenediamine. For your notebook this week, I'd like you to document and describe the results of each of the tests for your 4 organisms. One of the things we'd like you to do when you're taking pictures of these is actually, if you can just make a note card and put your name and the date on there in your picture, that would be helpful to us. With each of these, we'd like you to interpret what those results mean. So here you're kind of talking about whether it was positive or negative. Here you're telling us what that tells you, right, about the metabolism of the microorganism. And then 2 other things in here to explain the overall use of pH indicators, some of the things that are causing increases or decreases in pH, and then also the idea of the use of redox indicators, right, like in the catalase and oxidase tests. Okay. Any questions about that? All right, so that's the main thing. Inoculating these won't take you long. Don't forget to also inoculate new stocks to carry forward. There's nutrient broths there for you to do that too. Yes?
Are our stocks from 2 weeks ago still okay to use?
Yeah. Yeah, they should be. They're not going to be terribly healthy, but there should be enough viable microbes in there to get your new ones up and running. All right, I think that's it for the intro to this. I do want to talk just briefly about quantitative PCR. So we probably don't need that.